Maciej for this nice introduction. So today I will tell you a story of one of PhD of mine, it's Nina Preisner, and it's a combination of two actually stories. Her master thesis when she was working on antibiotics in different environments and checking them, and my story of polyhydroxyalkanoates, so bacterial polymers, and how to marry those stories. So today this is the presentation about. We decided when we were looking at one of the molecules, why not to combine the knowledge on the antibiotic resistance and the production of the polyhydroxyalkanoates. And why so? I'll come back later to this. We focus on beta-lactam antibiotics, and this is a group of very old antibiotics that have been launched into the market in the late 30s and early 40s of last century and saved millions of lives. And you can divide them into 5 groups: penicillins, cephalosporins, monobactams, penems, and carbapenems. And all of these antibiotics, they have those beta-lactam cores. In the case of penicillin, this is 6-amino penicillic acid, and they differ only with those appendixes in here. So when you change that little molecule, you can obtain a different range of different bioactivity of those molecules. These simple molecules work on destruction of the biosynthesis of a cell wall of gram-positive and gram-negative bacterial strains, working as a Trojan horse, exchanging this dipeptide, the-alanine, as they are structurally similar. So bacteria once encourage the antibiotic, halts its synthesis of the cell wall. And end of story. So coming back to the structure of the most simple and the first discovered antibiotic, penicillin G, we have two parts of the molecule, a beta-lactam core and this little appendix that resembles a PHA monomer of one of the polymers we can synthesize. So what are the PHAs? This is an abbreviation of polyhydroxyalkanoates, and this is a large family of biopolyesters. These polymers are accumulated by numerous strains in the nature of bacterial and archaea, and they are accumulated as little granules within the cell of bacterium. So bacteria accumulate these polymers in response to the environmental stress, so similarly to bears accumulating fat to survive the winter. But these polymers, PHAs, are a source of bioactive molecules or 3-hydroxy fatty acids. And up to now, over 150 different 3-hydroxy fatty acids have been identified that they can build those bacterial polyesters. Usually they are simple aliphatic monomers, but they can have also some really interesting groups attached to the end or within the alkyl chain of the monomer. So the purpose of the study was to actually work up the method of chemically or biocatalytically synthesize novel antibiotics based on the monomers and on an example of 6-amino penicillic acid. For that purpose, first of all, we needed to get some polymers in order to produce two distinct set of monomers. And we targeted an aliphatic polymer and a polymer with an aromatic group in the end that resembled this of penicillin G. In order to get the aliphatic polymer, we used fed batch culture, high cell density fermentation processes to obtain around 140 grams per liter of bacteria with 70% of a plastic inside. And as it is very cheap substrate, but in order to obtain that aromatic polymer, we used a pulse fed batch culture in flasks because it's the 5-phenylo valeric acid is very expensive and it's in solid form, so it was needed only small quantities. After both fermentation processes, bacteria accumulated the polymer as in strictest granules inside it, and then we use the process of getting out the polymer, and in the end we depolymerize it to obtain first substrate for the reaction. So methyl esters of aliphatic monomers and as well 3-hydroxy-5-valerate methyl ester. The next step was also to obtain a small library of fatty acids. So we used a very simple saponification with lithium hydroxide in order to demetallize and obtain the fatty acids themselves. And Justyna's PhD basically focused on developing of two pathways, as I said. First, a chemical one, which we use the substrate in its carboxylic form, and then the use of lipase in order to construct a library of differently substituted novel penicillins. But first of all, the first step was to convert 6-APA into a benzyl ester in order to enable smooth catalysis in one of the solvents of choice to make it more hydrophobic. Yes. So first, I will give you an example of the chemical pathway that she developed. So she has been testing a lot of condensing agents used in organic chemistry, and the best results were for this 3,3-P reagent in order to make possible new amide bond formation. And then in the end, over the palladium catalyst, Justyna was deprotecting the benzyl protected carboxylic group of the molecule in order to obtain biologically active molecules in their sodium salts. But we said to ourselves why we need to use this harsh organic chemistry in order to prepare the novel antibiotics, where we can tap into biocatalysis. And we used a very simple catalyst, lipase, in order to exchange the methyl group of the monomer with the 6-aminopenicillic β-lactam core. So Justyna first of all tested several different organic solvents and then tested around 30 different lipases. After that big screen, she concluded that the best enzymes are CalB, the commercial Novozyme enzyme, and TLEM as well. So we obtained really high yields for aliphatic and aromatic novel beta-lactams, but over the course of analysis and workup to clean up those molecules, she found out that they are very temperature labile. So she had to work in low temperatures for a longer period of time, and then be very careful when she was purifying those antibiotics. Doing so, she obtained a library of differently substituted novel beta-lactams. As you can see, these that were very similar to penicillin G, also the ones that they had different aliphatic lengths, changing their hydrophobicity as well, and this had also an impact on the biology. So now I will move to the biological results. There will be two tables with a lot of results. Just remember, red is bad, green is good. And Justyna tested these in two ways. First, she selected bacterial strains in Warsaw, gram-negative and gram-positive strains from whether the reference list or some clinical isolates. And also, then she moved to testing the cytotoxicity of those novel beta-lactams. As I said, a lot of things are happening in here, but just to tell you where to focus. First column is penicillin G, and we are looking at gram-positive bacteria test. So as you can see, most of our derivatives, they are working nicely or at the same level as they work with for the penicillin G on the several Staphylococcus, Enterobacteriaceae, especially Staph aureus and Staph epidermidis. When we change bacteria to gram-negative strains, only those of Proteus mirabilis and vulgaris, which were very sensitive to ampicillin in this case, gave very similar results with our antibiotics. So the last part of the research was to check the cytotoxicity of novel molecules, whether they can be substitutes for the drugs that are currently on the market. And we could show that when you compare different concentrations, low and high, to penicillin G, for example, they are very non-cytotoxic, especially when compared to ampicillin in the highest concentration. So in the end of the story, I would like to tell you that it is possible to use PHA monomers in order to create new molecules, especially antibiotics on this example. Those are very interesting molecules because bacteria can produce over 150 different monomers. But moreover to that, Wojtek is here also, he was modifying the 3-hydroxy group on the PHA monomers in order to introduce different biological activity by adding some halogenation onto it. And he was synthesizing actually the sugar esters. So this gate of 3-hydroxy group on the pH monomer opens even bigger routes to modify and discover new antibiotics that can in the future help us in the fight with bacteria. So thank you very much for this